Okay, so today's lesson is about hemic system diseases. Okay, so blood, this plays a vital role in maintaining the health of birds. Okay, so when we study hemic system diseases, we need to be acquainted with several things. Okay, that would include the normal values of hematological parameters in poultry. Okay, and then also, what are the common reasons for diseases of the immune system? Okay, you may include here anemia. Okay, anemia, which can be due to nutrition, infection, as well as parasites. And then another would be bleeding tendencies. Okay. And then another reason would be conditions which cause abnormal white blood cell counts and then disorders which would affect the bone marrow. Okay, like what I mentioned at the start of this lesson, okay, we need to know more about the normal avian hematology. Okay, so the total blood volume in clinically normal birds, that would be in the range of 6 to 11 ml per 100 grams of body weight. Okay, so for example, you have a bird weighing 250 grams. Okay, this bird would have approximately 15 to 27.5 ml of blood. Okay? In a clinically normal individual, you can draw up to 10%. Okay? That would mean about 1.5 up to 2.7 ml. Okay? Right. So the next question would be, where can you get blood from poultry? Okay? So the four key sites where you can get blood. This would include the brachial wing vein, the medial metatarsal vein, the jugular vein, as well as the heart. Okay. Bear in mind that the volume of blood that can be collected from each site. Okay, this will depend on the weight of the chicken, the skill of the phlebotomist, and the speed of blood clotting. Right. So when you collect the blood, the normal ranges of the hematological parameters in chickens would be RBC at 2.5 to 3.5 times 10 to the 6 microliters, PCV 22 to 35%, hemoglobin 7 to 13 gram per TL, and white blood cells 12 to 30 times 10 to the 3 microliters. Now, we've known about the some details about avian hematology. Let's now move to the diseases. Okay? For the first disease that we'll be talking about, this would be chicken infectious anemia. Okay? So, what is chicken infectious anemia? The other names that this disease is known as would be blue wing disease, okay, anemia dermatitis syndrome, also hemorrhagic aplastic anemia syndrome. Okay. So what it is, it's a viral disease of young chickens. Chickens that are aged 3 to 12 weeks. And it is characterized by chick mortality, plastic anemia, and then immune suppression. So the agent, okay, it's a chicken infectious anemia virus. Okay. So this virus is the only member of the genus Gyrovirus of the Anelloviridae family. Previously, this virus was classified as a circovirus, okay? But later on, it was found that important differences in genome organization led to a reclassification into this a new anellovirus family. Okay, so it's 25 nm non-envelope icosahedral virus with a single-stranded circular DNA genome. Okay, it is the smallest possessed by animal viruses. Okay, so the disease has an 8 to 10 day incubation period, and the principal sites of the virus replication, these are the hemocytoblasts in the bone marrow, precursor T cells in the cortex of the thymus, and also dividing CD4 and CD8 T cells in the spleen. Okay, so replication in and destruction of the hemocytoblasts, this would lead to anemia. Okay, now replication in and destruction of the T cells, this would cause immunosuppression. Okay, so neutralizing antibodies, they are detectable 21 days after infection, and clinical, hematologic, and pathologic parameters return to normal 35 days after infection. Okay, so here's a description. There are viral proteins, VP1, VP2, and VP3. Okay, so VP1, this is the only structural protein. This is the capsid protein. Okay. So VP2, this is needed as a scaffold protein to allow proper folding of VP1. Okay. This also has serine, threonine, and tyrosine protein phosphatase activities, and mutations affecting these activities result in the attenuation of virus replication in vivo. Okay. So VP3, these are non-structural proteins that induces apoptosis in infected cells and has other functions in viral replication. Okay, so the host would be maternal antibody negative chicks, which are susceptible to the infection and disease until 1 to 2 weeks of age. Okay. Now, for maternal antibody positive chicks, okay, these are protected from disease, and probably from infection. Okay. So, age resistance is related to clinical disease, but not the infection greater than 1 week of age. Okay. So, this disease is complicated by co-infection with the following. Okay, so Marex, okay, infectious bursal disease virus, and then reticuloendotheliosis virus. Now the transmission, there is vertical transmission. Okay, so seronegative hens become infected and continues until neutralizing antibodies develop. Okay. So roosters, they also shed the virus in the semen. And then horizontal transmission, this would include fecal-oral, okay, respiratory routes, as well as infected feather follicle epithelium. Okay. Also from viremic chicks. Okay. Now talking about immune suppression. Okay. So immune suppression has adverse effects on proliferative responses of splenic lymphocytes, production of interleukin-2 and interferons by splenocytes, and generation of antigen-specific cytotoxic T cells. Okay. Also has adverse effects on macrophage functions. Okay. So some clinical horizontally acquired infection with chicken infectious anemia virus in broiler. Okay. The progeny of seropositive parent flocks may be associated with impaired economic performance. Okay. Why so? Chicks would be observed as anorexic, lethargic, depressed, pale, with hemorrhaging. Okay. So signs of illness or adverse effects on egg production do not occur when seronegative adult chickens become infected. Okay, so the infection of maternal antibody negative chicks before 1 week of age, this can cause the clinical disease 12 to 17 days after hatching or infection. Now, checking for the hematological signs, there's anemia, hematocrit less than 27%. It's also leukopenia. Okay? So thrombocytopenia due to cytotoxic effect of virus on bone marrow precursor cells. Okay, there are also mononuclear inflammatory cells, which are eosinophilic or red internuclear inclusions. This can be found in thymus, spleen, bone marrow, bursa, and lung. Okay. Here, associated increased mortality, and then readings of the hematocrit to your right. And then you can see bleedings on the wing. Okay. So postmortem lesions, this would include pale bone marrow, severe thymic atrophy also of the bursa, and then hemorrhages in the skeletal muscles. Now, filling in the blanks, it's blank necrosis and depletion, and then bone marrow blank. Okay, so blank necrosis and depletion, that would be lymphoid. Now, the answer is lymphoid. Lymphoid necrosis and depletion. Then bone marrow blank, okay, bone marrow blank, that would be bone marrow panmyeloptysis. Okay, so panmyeloptysis is defined as the wasting or degeneration of the blood-forming elements of the bone marrow. Okay, so to diagnose the disease, we check for the history and signs, the growth, as well as the histopathologic lesion, and also the reduced hematocrit, thymic atrophy, and pale bone marrow. To confirm, we do this by detection of the virus or the viral DNA in the thymus or the bone marrow. Then there's viral isolation, but it is slow and expensive. Then commercial ELISA kits. Okay, this is used to identify breeder flocks that are seronegative prior to egg production. And then to monitor the efficacy of vaccination. Okay, so differential diagnosis would include diseases which cause immune suppression. Treatment, there would be no effective treatment, but you may still give antibiotics to prevent secondary bacterial infection. And then for chicks, increasing the brooding temperature, okay, may help reduce losses. And then vitamin C, okay, can also be provided. Now for prevention and control, okay, this would include medical prophylaxis, okay, vaccination of seronegative flocks prior to the onset of egg production is recommended to prevent vertical transmission. Addition of crude homogenates of tissues from affected chickens to the drinking water. Then there's also single vaccination prior to laying, which will provide your birds with lifetime immunity.